Officially starting. Thank you so much, Dr. Shaw, for being here with us. Welcome to the sounding board. Can everybody hear me? Can everybody hear me? Yeah, thank you very much. I'm the sounding board chair. This is Oceanids 68th year, which is an official support group of the university. The sounding board is a joint effort between the faculty club and Oceanids. Our honorary chair is Despin Kabulakis. She's the associate of the chancellor, and our president is Nigella Hilgard. In Oceanids, we raise money for grants and awards for students, support other UCSD events and activities such as the student-run free clinic. We've offered many events for our members, and the sounding board is one of these events. We would love to have you as a member, and I encourage you to join. It's only $35 per year. We are very happy to see you now via Zoom. I guess we are getting used to it now. We are here every first Thursday of the month and throughout the academic year, except for January. Now, I would like to introduce to you our speaker, Dr. Reuben Shaw. He is the director and professor of molecular and cell biology laboratory at the Salk Institute, NCI Designated Cancer Center. He's also the UCSD Adjunct Professor of Molecular Biology of the Medical School. And a little about Dr. Shaw. Dr. Shaw got his Ph.D. at MIT and did postdoctoral training at Harvard Medical School before starting as an assistant professor at the Salk in 2006. He was the recipient of several Young Investigator Awards, including the Howard Hughes Medical Institute Early Career Scientist Award and the NCI Outstanding Investigator Award. Research in the Shaw Lab and the Salk Institute focuses on the LKB1 AMPK pathway, a central energy sensing pathway in all cells that reprograms metabolism in low nutrient conditions, and which is altered in a large percentage of lung cancers. Decoding this cellular starvation pathway has led to many new therapeutic targets in cancer and metabolic disease. The subject of his presentation is decoding tumors' weaknesses to develop better therapeutics. Please go ahead, Dr. Shaw.
Great. Thanks for that, Nellie. Thanks, everybody. This seems like a really fun group. So it's an honor to be with you this morning. I hope everybody has their beverage of choice as we're at lunchtime here. What I'm going to tell you guys about today is about a central focus of my laboratory's research. But I'm going to try to have this be more of a conversation and try to talk about this in a way that if anybody has questions, and if I start talking in too many scientific terms, please just yell out and I'll try to rephrase and just having the benefit of talking to you, hearing from people for the last 10 minutes has made me realize that this can be a very nice interactive opportunity. And rather than going through some of the science slides that I have in the slide deck, I will try to really bring this to a level where everybody can understand what the main points are and we don't get too hung up on all the scientific jargon and what the different details all are. So as Nellie just said, I'm a cancer researcher who is a straight PhD, so I don't see patients and I have no medical training. I was an undergrad at Cornell in upstate New York. I grew up in upstate New York and had the good fortune to work in a research lab as an undergrad. And that's when I first learned about genes and about sequences within our DNA that determine all different features from our hair color to our eye color to whether or not we're predisposed to diabetes or autoimmune disease or cancer, neurological disorders, all of that happened to be coming to light, and scientists were really just first discovering those genes that cause these different long known diseases that have been known for millennia. And they were actually finding the true cause of those diseases in the mid 80s and early 90s. And so I happened to be an undergrad at that time, and I couldn't believe the things that were being discovered that were not yet in the textbook. And I just fell in love with doing research, and it led me into cancer research and these more difficult problems to tackle. And maybe one of the themes that I'll tell you about today is that even though my training and focus has kind of always been centered on cancer and coming up with new therapeutics and ways to attack and defeat cancer, along the way, we have discovered that all of the diseases I just mentioned are far more intersecting, and the actual genes and some of the biochemical pathways and processes they regulate are actually far more related to each other than you would imagine. And so some of the things that I'll tell you at the very end, like the synopsis, is really that some of the cancer genes and things that are altered that give rise to cancers in our bodies are also controlling some of the same processes that go wrong in different common forms of neurodegenerative diseases such as Parkinson's and Alzheimer's, and also underpin some of the changes that occur in metabolic diseases such as diabetes. So one of the ways that cancer researchers have really tried to frame a general understanding about how cancer forms and about, what goes wrong. How does a cancer form in the first place? And then what are different changes that occur inside the tumor cells that allow it to survive and grow in our bodies and take over parts of our bodies. And one of the frameworks that they came up with, and by they, I'm talking the general, tens of thousands of cancer researchers, but what some of the leaders in these fields in the United States and around the world came up with or agreed upon is that we would call some of the common features, the few things that are in common across all of the hundreds of types of cancer, we would call them the hallmarks, these critical hallmark features of cancer. And so in some very important and influential review articles, some professors at MIT and at UCSF laid out this framework, which then many others have adapted to or have adapted, and it's become kind of codified as a general framework for thinking about cancer. And there are really 10, which depending on your perspective, that sounds like a lot, or that doesn't sound like that many, but there are really 10 specific steps that kind of go wrong inside the tumor cells in every kind of cancer that comes up. Now, sometimes multiple of these steps go wrong at the same time. So it's not like 10 isolated individual things have to happen bad. Sometimes one bad particular thing that happens causes like 4 of these particular changes to occur. But this is really a way of thinking about it. The main thing is that normally all the cells in our body live for a certain amount of time. Some cells, like cells in our brain, actually live for many of the full length of our lives. So that, the individual cells can actually stay alive for decades. Other kinds of cells, like cells in our bodies that are exposed to the environment, including our skin, including the lining of our gastrointestinal tract or digestive tract, and including our immune system that is constantly bombarded by things we breathe in, those cells naturally turn over. They are created, they live, and they die all within a day or two. So the whole time we are alive, the surface of our skin, when you have dry skin, you scrape your skin like I am right now, those are cells that were born sometime in the last couple of days. They lived, they absorbed energy and things from the sun and whatever I was exposed to. And then they naturally pass away. The cells die, they slough off, and new cells come up right behind them. And so the health of our skin is actually based on the fact that the cells never live that long. They're constantly actually naturally turning over our entire lifetime. And so, but if a skin cell didn't actually slough off when it's supposed to, that could be the first step that helps to lead that cell into turning into a little benign cancer. And so the actual most common form of cancer, or at least we wouldn't normally use the word cancer because it rarely gets bad, but it truly is an overgrowth, is actually something called basal cell carcinoma. Most people in their lifetimes actually get at least one basal cell carcinoma. You wouldn't even know it if you haven't had it. I probably have one right now. Like, it's generally just a little rough patch of cells, often on your forehead or some area of your face that's exposed to the sun a lot. Happens a lot on people's faces. Obviously, it can also happen on your arms or your legs, depending on how exposed you are over time. And this, in and of itself, is not a bad thing. Basal cell carcinomas rarely become so aggressive or dangerous that they can't simply be removed in what is often just an outpatient procedure in your dermatologist, or occasionally even your primary care physician's office. So the ability of cells to live for a longer time, or even for small tumors to come up in our bodies, is not by itself a dangerous or horrible thing, as are some of the deadliest forms of cancer that can start within our bodies, can take over whole vital organ systems, and we have no idea that they're there. And then when we have them, seemingly our bodies are not in our control, and these tumor cells grow and they can't be stopped. And so these other things that go wrong inside those cancer cells, and depending on what tissue it started in, that dictates everything else that happens next. Is this the type of cancer that surgeons or clinicians have the ability to try to physically remove? Is this the kind of cancer that responds to a conventional chemotherapeutic, a chemical that kills cells that are dividing? Or is this something that, modern scientists have invented a special medicine that only impacts what went wrong in that particular subtype of a subtype of melanoma, or colon cancer, or lung cancer, for example? So this is really just a framework for thinking about this. One of the things that goes wrong in many of these cancers, and is a feature of all cancer cells, is something called deregulation of cellular metabolism. And this is going to be the main thing that I focus on for much of the rest of the talk. So metabolism is simply a way of describing how cells take up nutrients from our blood. So when we eat food, the food enters into our stomach, enters our mouth, our throat, our stomach, gets broken down into all the little particles. So it's broken down into glucose and different sugars. It's broken down into amino acids and different, and then it's broken down into lipids, little fats and little components that then make up all of the other structures and things in our bodies, in our cells. And so we need the nutrients from food to do that. You can take vitamin supplements. Those are also things that are naturally found in many of our foods, but at certain age and things, it helps to take vitamins to keep your body healthy and to keep certain processes, running on time. So, but metabolism simply refers to the overall series of biochemical reactions, if you will. Biochemical pathways is a word we often use in science these days, that convert one type of sugar into another, that tell the cell whether it should be storing nutrients or not. Like if you just ate a big meal, your body actually releases a hormone from your pancreas called insulin. Many of you probably know this. It goes out and basically tells the rest of your body, hey, we just ate a whole bunch of food. And so it tells individual cells, start storing away the food you just ate. Like you just ate a meal, so it's time to like save those nutrients. Use some of them to do something good, like burn energy, exercise, or run. It allows us to have energy to power our muscles. It allows us to power our brains, but it also allows us to basically store that nutrient for another day. And so metabolism is that process. One way that cancer scientists, oncologists, and cancer researchers classify tumors nowadays are by changes in these particular pathways, but then on a more granular level, more scientific level, they actually look at the specific genes that are altered in an individual person's cancer. So depending on what cancer you or a loved one may develop in your life, when you go in, if they obtain a blood sample or a biopsy of your tumor, a very common thing nowadays is that they will send it out to sequence the actual genes that got altered in your cancer. Not just in skin cancer in general, or lung cancer, or leukemia in general. They want to actually know exactly what got changed in the cancer they just took out of your body. And the reason they want to do that is because only in the past 20 years, scientists have developed new, smart, rational drugs that attack the specific genes that go wrong in different forms of cancer. So that's really the good news. The good news is that for the first time in human history, scientists have understood how different forms of cancer start in the first place and have developed like the highest level of perfect drugs that can work to try to reverse what went wrong in the cancer. Some of those drugs today have put certain forms of cancer into what you would call permanent remission. They have put certain forms of leukemia, for example. Chronic myelogenous leukemia used to be a death sentence. And if people found out that they had it, they went from a day of having some modest symptoms, going to the doctor not feeling well. If they did a biopsy, took a blood sample, told them they had chronic myelogenous leukemia, as opposed to one of the other kinds of leukemia, they probably had about 18 months to live. And starting about 18 years ago, a new drug was invented and then tested for a decade, as is usually done in these cases. And that led to a way to treat that specific subtype of leukemia, because the drug called Gleevec directly binds to and blocks the single gene that actually causes that type of cancer. Now, that is just our first and best example of what is possible for the future of cancer research. So that's the good news. The complicating or the bad news is there are about 350 different genes that get altered in human cancer. And today, scientists have really good drugs for maybe about 10 of the 350. So we have got a long road to go. But the good news is we know what we need to do now in a way that we didn't for most of all of human history until the last 20 years. So my lab focuses on a handful of cancer genes that are altered in a couple different types of cancer. They're most commonly altered in lung cancer. They're sometimes altered in colon cancer and in pancreatic cancer and in endometrial tumors specifically. There's some specific thing we don't understand about uterine and endometrial tumors that they also share in common with these lung cancers and colon cancers. But that also tells you something else that may have been unexpected, which is that the genes that actually cause the tumors to form in the first place, they are not specific for a particular tissue. The same type of genetic event can happen in one cell of your lung that also happens in a cell in your colon, and either one of them will cause that tissue. But if that same thing happens in your skin, you won't get cancer. So it's really a statistical game as to whether or not any particular thing that happens could give rise to cancer. But again, the good news is that we are really boring down to a very atomic level to try to come up with new therapeutics. So the long story short, the upshot of my whole talk and the focus of my lab for all of these years has been a new cancer gene that was discovered in the late 90s when I was finishing up my PhD at MIT, was identified. It ran in families. It caused inherited forms of colon cancer. But the weird, surprising part was the same gene also was found very frequently altered in lung cancers, mostly in smokers, but sometimes in non-smokers as well. If you checked the sequence of their tumor DNA and compared it to their normal lung or anyone else's normal lung, it was very clear there had been a very specific change in this particular gene called LKB1. And so starting then in the late 1990s, I set out to try to figure out what does this commonly altered gene in cancer normally do, and what can we come up with to try to treat these specific patients. It's not going to help lung cancer patients with an alteration in a different gene. It's not going to help the colon cancer, endometrial cancer patients with things in a different gene. But with this concept that we're going to kind of pick off the bad cancers one by one based on their genes, that's what I set out to do. And so the big surprise from my training period was that I found, much to my own surprise and definitely to the world's surprise, that the major thing that that gene was doing was actually regulating a central process in the cell of how cells use and store glucose. And that was really surprising because people in the molecular, kind of the modern understanding of cancer field had really thought metabolism was not so important in cancer. And it wasn't because they just didn't know. It's actually because this had been one of the prevailing hypotheses as to what the cause of cancer was way back in the 1920s. There was a famous physician named Otto Warburg who was in Germany during the time of the First and Second World Wars. He won the Nobel Prize in the early 1930s. He was a luminary who came up with new methods and new ways of studying different things that relate to cancer. But this is before people knew about DNA. It's before they knew about genes. It's before they understood how things in our environment cause changes in our DNA. And they didn't understand what the actual cause of cancers was. So what Warburg was able to understand, though, was that there was something really weird happening in the chunks of tumors that he studied compared to a chunk of normal tissue. And what it was that the tumor cells were actually taking up way more glucose, sugar. They could measure the sugar in the media that they put a piece of tumor in, and they put a slice of normal liver, a slice of liver cancer, and the cancer cells were taking up way more sugar than the normal cells. And so Warburg noticed that, did a series of experiments to try to figure out why they might be doing that, and then figured in the end something was wrong with the way the tumor cells were normally using sugars. Sugars normally get into a structure in our cell called the mitochondria. On the next slides, it's thought as being the powerhouse of the cell. It's where all the energy for the cell is made from the sugar and the things we take into our bodies. And so Warburg thought maybe that's it. The problem was 80 years later, 70 years later, in the mid 1980s, around the time I was telling you that I was an undergraduate, scientists discovered that in fact there were specific genes that were changed in different people, both in families that were predisposed to cancer, but then when you looked in people who just randomly developed a type of cancer, the same genetic changes happened in their tumor cells, which for the first time really provided a smoking gun. And then when techniques got even more advanced and scientists learned how to change DNA in the germline of different model species and this happened right at the time that I was starting in grad school. In early 1990, scientists developed methods to be able to make changes in the DNA of mice so that the mice actually could recapitulate different forms of human disease, forms of cancer, but also cystic fibrosis, Tay-Sachs disease, you name it. And so the ability to do that genetic and genomic engineering of animals to mimic what happens in human diseases had never happened. No one had the ability to do that until around the year 1990. So then when they did, they engineered mice to have specific mutations like were found in the human patients. And the incredible part was that the mice developed cancer similar to some of the human patients. And so when scientists made that observation, it suddenly became very clear that even though other things could be going on, like viruses can cause rare forms of cancer, that is true. Maybe other changes in the diet can make odds of cancer increased, but that the real thing that has to happen is you actually have to have a genetic change inside one cell in your body that starts the process. You have to have something go wrong in one of those cancer genes. And so when that became clear that these cancer genes were really the true source of what caused cancer, all the other prevailing hypotheses that had accumulated over human history for what caused cancer suddenly went out the window. And I mean, went out the window really fast. So all the people who had held the microphone at the previous cancer meeting, big cancer meeting saying all over the world, the cause of cancer may be changes in metabolism and the structures and these cellular structures, all those people were basically run out of town by the people who had identified the genes causing cancer, because they were basically like, oh no, take that whole stuff about glucose, all that malarkey, and get out of town, because now we know the true cause of cancer, and it's these genes. So when 20 years after the discovery of cancer genes, I found myself that one of the more common genes altered in human cancer turns out to regulate glucose. I was actually not very excited that day, even though I knew the very day I discovered it, this was going to be a huge finding. I was not so excited because I thought, oh no, like these people hate this idea. Like this isn't actually a new idea. This is a really old idea. And they're actually going to hate that I discovered that this cancer gene, the main thing it does is regulates glucose. Like they're not going to be happy. They're going to be like running me out of town. And that is actually kind of what happened. Like it took a couple of years, and it took other people to repeat everything that I did for them to believe it. But obviously I've been more in storyteller mode than I have been actually like giving you my slides, but hopefully that's maybe better appreciated for this particular audience. What I do want to do though is jump to just a couple of the slides that I prepared to kind of tell you a few key things, because I want it to allow time for us to talk and not just for me to dominate the microphone here. So the long story short of what this particular kinase, LKB1, normally does is that it serves as a sensor in all the cells of our body for how many nutrients, like the overall health of the cell. And one of the ways our cells read out whether or not they have enough energy is through this molecule called ATP. It is the most basic fundamental building block of all living things. And it powers all the little machines in our cells, all the DNA, all the RNA, all the enzymes, all of them need ATP to basically do their jobs. And ATP gets used up by all of these things. And when it does, it gets converted into a smaller form of itself called AMP or ADP. The way this particular cancer fighting enzyme works is that it is actually sensing the pure absolute levels of AMP, one of the breakdown products in the cell. And when the levels get too high in the cell, it is very much like a one of those signs that says, in case of emergency, break glass. If the ATP levels within the cell get too low, the cell activates this kinase LKB1, and then it's an enzyme. It gets flipped on and it sets into motion a series of biochemical reactions that tell the cell, we have got a major problem. We have got to turn off all the major engines of the cell that are using energy, and we basically have to go on auxiliary power until we figure out what the big problem is. And so it's actually a network of enzymes and proteins that work together to figure out where the power line went down in the storm. And then they send out, literally just like you would, like this analogy, you send out actually a crew to the part of the cell to try to repair where the power line went down. And if you can't repair the power line, they send a signal back, and the cell actually naturally kills itself, is so to not stay around with low energy and potentially like turn into a cancer someday. It's actually a really complex but decision-making series of enzymes and apparatus within our cells. And another way of thinking about it is it's really like a fuel gauge. If your cell gets too low, this enzyme gets activated. If it gets too low on gas, this enzyme gets activated, but it'd be like a self-driving car. Like if your car, when it got low on gas, automatically drove itself to the gas station, that's kind of what this enzyme does. It changes everything so that it would turn off your radio, turn off the windshield wipers, turn off the air conditioning, and then drive the car to a gas station. This enzyme tries to basically restore normal energy levels. And so over the last 16 years now, since I moved to San Diego, we have been focused on all the little details of how this one enzyme kind of accomplishes all these miraculous things. Along the way, we discovered that some really ancient, pro-health interventions, such as exercise, in fact, normally work. Part of the reason that they actually have health benefits is that exercise naturally stimulates this anti-cancer drug. And another unexpected thing was that the widely used frontline type 2 diabetes drug Metformin, that some large percentage of the population take, actually also stimulates this same anti-cancer drug. Or not anti-cancer drug, this anti-cancer enzyme, LKB1, and it's the thing that does all its benefits. LKB1 just flips a switch and turns on this enzyme AMPK that is written in the middle. And then AMPK basically is the messenger that passes it out to all the targets throughout the cell. And so this has been a central focus of my lab. We, in contrast to how this may sound, if you hadn't heard any of this before, I'm actually naturally a skeptic as a scientist. So when another scientist tells me something like, oh, hey, I think I found that diabetes drug is also regulating my favorite enzyme. And turns out exercise does it too. I'm like, get out of here. Like, just get out of town, man. Like, that sounds too good to be true. That's like absurd. Like, what else? Does sunshine activate it? Does your favorite beer also turn it on? And of course, sunshine nor your favorite beer turns it on. So it really is specific to both exercise and metformin. Lots of other beneficial things do not turn it on, but it turns out that exercise and metformin turn it on. What we could not have known, though, is that by studying this pathway, and now I'm going to jump through a whole bunch of science slides that you don't need to know, but what it does, what AMPK does to basically restore the cell to health in response to exercise or metformin, it is basically putting back together. It's like Humpty Dumpty. It is basically putting back together all these cellular structures, like this mitochondria shown on the upper left-hand side there. That's actually a little floating factory within all the cells of our body that make ATP. It makes sense. The number one place, like in a blizzard, that like where power lines could go down is probably, if all the lines are up in the air near the power station, probably half the lines go down from trees near the power station. And that is exactly like what a mitochondria is. It is a power station of the cell where all the other energy flows out of. And so these molecules, LKB1 and AMPK, these enzymes, they guard the power station, because if anything goes wrong at the power station, the cell is in serious trouble. So, so that's one of the things we discovered. And there's all different little biochemical details of how it does that. It also tells the cell, one of the other surprising things we found just in the past year is that one of the ways it helps make cells healthy is that it actually directly regulates another gene and an enzyme well studied for a completely different disease. And this was another surprise, which to me is just the joy of science, is discovering things that you can't even imagine. It turns out that this LKB1 AMPK pathway directly catalyzes a previously unknown thing in the gene that causes early onset Parkinson's disease. So Michael J. Fox is obviously a famous actor who has early onset Parkinson's disease. Many people, I've had people in my family, many people know someone who has either late onset Parkinson's or early onset. Almost all of those cases are caused, just as cancers are caused by changes in specific genes, they are caused by changes in this gene Parkin. And what we discovered is that our pathway that fights cancer, that fights type 2 diabetes, turns on the ability of this particular enzyme to also serve as a barrier and a barricade to developing Parkinson's. So my final slide is that in my 20 years since I was first trained of being a scientist, I started off here working on cancer genes and how cancer forms, found that in fact, these things are regulating glucose and they're regulating lipids and they're sensing glucose and lipids. And when things are bad within the cell, this single gene turns on this other enzyme that goes around catalyzing these reactions that fight diabetes and fight Parkinson's. And at the same time, it's fighting cancer. So I think we are coming up with, for the first time, in scientific history, a general framework for understanding how diet and exercise and the normal health of our cells and bodies are so intertwined. And with that, I will say that we have another thing I will not talk about today, but maybe a different time. We come up with therapeutics based on these new things that we find, the details, the molecular and atomic details of the enzymes catalyzing these steps. So I want to just go to the picture of the people in my lab who did the work. If I can find, there we go. Yeah, so this is the largest my lab ever got at one particular point, but we generally have about 15 different people, PhD students, undergrads from UCSD, PhD students from UCSD, and I teach. I give lectures on general cancer biology, the history of cancer metabolism, and on different steps. I also lecture to some of the medical students in endocrinology and how hormones affect metabolism. And with that, I will love to take conversations, take questions, or have conversations about any of the crazy stuff I just told you. Thank you.
Thank you so much, Dr. Shaw. Would you like to stop sharing your screen so we can see the audience? And
I had a question.
go ahead, Jen.
Sure.
You mentioned genetics and genomics. What is the
Yeah, that's a great question. I really like when people ask questions, because even when I'm trying to not use jargon, it's just so, it's scientists, it's like, it's like engineers or any specialist in something.
Right.
Business people start slipping into their fiduciary fund, or I'm like, I don't even know what that is. Is that a thing? Is that an adjective? Like in this case, genomics is more a term that means you're studying the actual sequences of the DNA itself. And it usually refers to that kind of studying right down to the changes that happen within the DNA. Genetics is related. Genomics, in other words, would kind of be like one subspecialty of genetics. Genetics is more studying any of the changes, they are all almost always in DNA. So it almost always would involve genomics, but it's more generally just how things are inherited and how much different traits in animals or plants or people, pass over generations. Genetics is really more the study of traits over generations. The study of that is called genetics. And then one specialty within that is how much of that boils down to these changes within the DNA sequences, and you would call that genomics.
Thank you.
Sure, great question.
So I have a question,
Sure,
Marilyn.
Marilyn, yeah.
Hi, Reuben.
Hey.
The thought is that maybe we should all be taking a little bit of metformin, like with our vitamins. What do you think about that? Because now you're saying it's going to prevent Parkinson's. God
Right.
knows what else? Maybe Alzheimer. Have you looked at Alzheimer?
That's right. No, actually, that's a great question. First of all, this is a fantastic question, because that is, from a scientific basic, at the basic discovery level, that is what these results suggest. No one has ever tested that in a broad way in the general public in people who don't have type 2 diabetes. However, because so many people do have prediabetes or metabolic or type 2 diabetes itself, there are actually millions of people who are taking metformin and take it every day for a long time also. So it doesn't have the kind of side effects that we normally see with any cancer treatment. Usually has some pretty horrible side effects. So metformin, for the most part, is safe. Often when people start taking it, it makes your stomach upset for like the first 10 days or something in like half the people. But then oddly enough, it kind of bounces back out. Which
years ago, James Watson came here to Salk and gave with his prostate cancer, and he was saying, I remember that. Oh, you guys, just take metformin.
That's right. That's right. A lot of people, I would say, Marilyn, a lot of people are. Oddly enough, I am not. But I've really thought about it. Like in the last couple of years, I've really thought, like, why am I not?
Yeah,
I don't have a great reason.
Okay.
Honest, I really don't have a great reason why not. I'm more of a trepidatious person in general.
Have you been vaccinated against the COVID then?
Yes, I have had all of my COVID. It all depends on it's the level of threat, right? Like it's the level of like something real bad's going to happen real soon. I will take the medicine.
Or you won't be able to come to work.
I want to hold off bad things and take medicine. Like I don't know. I'm also not good about taking vitamins, even though there's no question they would be good for me. I turned 50.
Do you eat food, or do you go on this caloric restriction diet?
No, I don't have the willpower to stay on it. It'd be neat if you did. And some people take it too far. You definitely can take it too far. My old boss from Harvard is actually quite famous now. He's been on 60 Minutes, very much about how sugar. I made some of these discoveries in his lab, and he loved it. And he's world famous, a cancer biologist named Lewis Cantley. And he has been a prominent person now saying, cut all sugar out of your diet and things like, sugar fuels cancer. And that may be true, but like cutting all sugar out of your diet, to me, unless there's another more acute medical reason to do that, that is just a very extreme response. You can get cancer many other ways. Cutting sugar out of your diet is going to have much bigger consequences. So I'm more of the moderation within diet. But coming back to metformin, I really do think it is possible, not like in 50 years, it's possible even within 10 years that this may turn into something much more like baby aspirin, or something where people do routinely, that the government, in other words, finishes clinical trials in the general population and decides that there is enough of a benefit. Now, one of the reasons metformin, though, won't actually mimic all of the benefits of some of these things is that, and this is a good thing, this is actually why it doesn't have so many side effects. Metformin mostly is able to penetrate a couple tissues in our body. It gets into the liver really good. That's where that's why it helps so much with diabetes, is because the liver is where you uptake glucose, and in it, the liver sends a signal to the pancreas about insulin. So it works really well for that. It also gets into your GI tract. So actually, for people with colon cancer, metformin may even turn out to be a therapeutic in that type of a setting. But the other thing about it, which is why it's so safe, is it's not that strong. It's not that potent of a drug. You know, modern molecular drugs like the Gleevec drug that like conquered CML, it is a really tight binding nanomolar machine that gets right into there. Metformin is like a weak thing that is almost like what it really is it is like a defibrillator to your mitochondria. It isn't actually damaging that much, but metformin is actually a derivative of a natural product that is normally found in a species of lilac, believe it or not, something called the French lilac. This is a true story. This is how metformin was actually engineered. I can't believe actually that no one has made like a greatest selling novel or a screenplay out of this, because it sounds like other movies, but this is actually the truth. This is literally a derivative that comes out of this one particular lilac species, which I'll add is actually pretty common. It's like an ornamental called French lilac. This particular one in the plants it was known back in the Middle Ages that it had medicinal properties, and people would chew on the leaves or the stem, and it caused animals to faint if their blood glucose got too low. But the animals had to eat a lot of it. And so anyways, that one of its other names from Old English was goat's rue, R-U-E, because like goats, I guess, particularly like the taste of it or something, they would eat so much that they would actually faint. But in people, if you took too high of a dose, but everybody knows. So 500 milligrams, someone asked, 500 milligrams is the common dose that actually is effective at doing most of the benefits of metformin. And I would not advocate needing to take more than 500 milligrams for most of the good things that metformin does. Some people, when their diabetes gets bad, doctors can prescribe up to 1,000 milligrams of metformin, and that usually impacts people more. But we don't know if there's any downside of taking 1,000 for a long time, whereas for 500, we know it's generally not a problem. So if I had to give offhand advice, I would say 500 is safe. But of course,
can I ask a question about that?
Yeah, sure.
So as you said, millions of people are taking it, so the side effects must be really minimal then.
That's right.
And if somebody has low blood sugar, it might not be a good idea though. I mean, it sounds like that's one thing you can kind of
yes,
run into trouble with your blood sugar.
That is true, but your primary care physician would even be able to know if the reason your blood glucose is normally low, if this would combine with that or not. In other words, this actually is not a general blood, it's not a general blood lowering mechanism. There are many people, and actually the many people who take it who don't have type 2 diabetes, it actually doesn't generally lower their blood glucose very much at all. The reason it works so well in type 2 diabetics is that one of the ways they have too much glucose specifically is that their insulin sensing pathway is off, and they basically don't just have normal glucose that all of us have. They have like this extra glucose that has come in, and so the metformin is really helping clear all that extra glucose, but it will not drive it that particularly low. Like metformin almost never can kill someone, for example. There are more potent forms of metformin that were used and were prescribed for type 2 diabetes in the past, one called fenformin, PHEN-formin. That's still actually used in Italy and in Brazil for type 2 diabetes. In the United States, it was pulled in the mid-70s because if you take too much of it, it can cause your blood glucose to get so low that it will kill you. Metformin almost cannot do that. Even if you accidentally took 6 of them, it just would not kill you very fast. So you would go to the hospital, it's just not as effective, which is why it's so safe. And it's why it doesn't have other side effects. One thing I will tell you, if anyone is taking it, is that for any reason you're taking metformin, the one, besides having a upset stomach for the first week that you start taking it, the other very common known side effect is it affects your vitamin B12 levels. So you just have to either supplement with vitamin B12 or just make sure that your doctor checks that out after you've been on it.
It's 1 o'clock.
So I have.
Or if you're a vegan, because the vitamin B12 is, you don't get very much if you're a vegan. If you can watch that.
Yeah.
Yeah, I have just one more question.
Wait,
Reuben.
yeah, Nona's got her hand up. I think
Oh,
she's had it up for a
Go ahead,
while.
Nona.
Sorry. Go ahead.
First of all, a comment. You're a moderate person and an intelligent person and gave a brilliant lecture, but you mentioned exercise several times, but it is not, we still de-emphasize things that people could do without taking metformin, which may prove to be a wonder drug. It is, you know, I am a family doctor, but I think people should be allowed the option. I think it should, like Marilyn mentioned intelligently, what about the semi-fasting diet?
Right.
There are extreme points of view, and my profession and your science always thinks the moderate person is the intelligent person, and they often are. And people like me that are headstrong and started the first preventive medicine center in Canada and almost got killed because it was a threat to McDonald's.
Yeah.
We're talking about the early 70s.
Wow.
I mean, you know, it was a threat, but being moderate is still an intelligent approach, but people should be allowed and encouraged. And we don't even know how much exercise.
Yeah.
You're still with metformin being 500 is probably, many patients hate being on metformin more than you let on.
I don't have enough personal experience, actually.
Your being moderate is wonderful, but people should have the option to do fasting and exercise crazily.
Oh no, I don't want you in any way to think, I actually think being moderate is probably not the best thing to do. I don't want anyone to think, I'm more telling you just like my own my personality. I for sure feel strongly actually that exercise, and I completely agree with you, as you know, as a scientist on the edge of the exercise physiology field, I have never heard anybody give me a succinct answer to, I don't think anybody really knows the minimal amounts of what type of exercise give different kinds of health benefits. Like the world just doesn't know yet. But what I do feel like the world does know is that exercise, more than any drug, is actually giving way more health benefits than even a perfect metformin. It's not possible because
I mean, even a recent breast cancer symposium hardly mentioned exercise as a reduction of breast. I mean, it mentions it now, but it's still not. I'm still, the emphasis is still on what, you know, I mean, metformin is a very cheap drug. And if it works as well as you say, it certainly would be wonderful to put it in the drinking water.
Sure. But metformin also will never, even though it has some of these benefits, I also want to be clear, if you have an alteration, a particular cell that's like going to drive lung cancer or something else, metformin is not going to completely stop any cancer or any diabetes. It's not that strong specific medication. It is more like modestly preventative. It's not as good as exercise. And people today, if metformin became as common as like baby aspirin, people will still continue to develop cancers and develop Alzheimer's and all kinds of things. So it is very far from being of deep impact. It's more just interesting in a way how this natural product derivative turns out to even have the benefits it does that people didn't know. You know, just like aspirin, like when people first were figuring out how it even works at all. It's fascinating to me that there still are drugs like this out there, other natural medicines that we have no idea why they impact the body and physiology like they do.
Anyway, thank you.
Yeah, no, thank you.
Can I ask another question?
Yeah, I can say.
Thank you so much for your presentation, because I used to be in the cancer field back in the 80s, and then I went into something completely different. And I'm shocked at how hard it was for me to actually look at your slides and everything, but your talk was excellent. I really appreciated it. I wanted to ask, going way back to your first slide about the 10 hallmarks of cancer, because none of that stuff was known in the 80s. And the thing that impresses me is that in the 80s, people used to say, you know, getting a bad sunburn when you're a kid or smoking or something is one of the steps. And there are all these other steps to cancer. And that's why you sometimes don't get cancer until late in life and all of this stuff. And these are the 10 hallmarks. These are the steps, right? I mean, this is it. So do you, can you say something more just in general about it? Because I'm so out of the field about how important these steps are. And I imagine you just focused on the metabolism step. There are the same amount of information and research and enzymes and everything behind all these.
Totally. Yeah. In fact, for some of the others, there's way more information than there is about the metabolism. Metabolism happens to be the one that I was focusing on, but even for me to make the impact that I did as, you know, as a younger scientist, it means that there wasn't that much out there other than the much older stuff. Some of these others have been much more in the sights of people, even in the 80s, how cells proliferate, the machinery that drives the cell cycle, that drives the cells to actually, and then all the machinery to make DNA separate and to divide and to have 2 copies of everything within the cell as it divides. All of those details, which also is one of the things that goes wrong in cancer, those are deeply studied. Those are, you know, giant, thick medical textbook relative, you know, amount of information. And so, and in those cases, of course, then we know more about the enzymes involved and the procedures of what they do. So scientists, we can come up with better drugs against those first. So it's also just always a matter of like, what was discovered first, and then what did someone find, you know, either through a rational, computational, big scale approach at a company or in a big research lab, or they just happened to discover something weird, like, you know, there's a fungus growing on this plate of cells, but it seems to be only killing the, you know, the lung tumor cells, and it's not killing the other cells. Like, that's weird. Maybe the fungus is making something. Like, you'd be surprised. You know, serendipity within the scientific discovery process is still very much, you know, that's still 10% of the things that turn into like big fields today. So yeah, I don't know if I even answered your question, but a lot is known about those 10 hallmarks now, and being able to group like individual, like I told you, there are about 350 genes, cancer genes total, and kind of lumping them into which one of these 10 hallmarks they mostly relate to. That's a whole nother interesting area.
Well, it's really exciting. It's a great time to be doing science.
Yes.
Thank you so much.
Thank you. Thanks to everybody.
Dr. Shaw, no pressure, but when you mentioned that we know more about 10 types of cancer and we're still missing the 340.
Yeah,
So,
I didn't mean to be depressing at the beginning. You'll notice I said that in words. I didn't want to put that on the slide. You can't start off a talk, you know, with that on the slide. Most people are just like, oh, you know what? This lunch was already getting rough. I don't have time for this.
What's the expectation? Are we going to be here to see that, or are we moving faster now that you have done all these advances?
Yeah, right. My straight, honest prediction, right? My best guess is that scientists now are moving fast enough that instead of discovering good treatments for one subtype of cancer every year or two, instead the advances are coming more every couple of months. We're discovering an advance for one of the subtypes of cancer. What that means then is that within 10 years, we're still not going to have treatments for half of them, but we are going to have treatments for maybe a third of them, at least a quarter of them, good treatments, right? Then I'm also in describing that to you with using these numbers of the 300 and 350 kinds of cancer, we're also not putting into that quantity how common some of them are. So another way of putting it is most people actually develop, one of 5 common kinds of cancer. There are another 300 kinds of cancer that we have a loved one or something are like, what? Biliary subtype cholangiocarcinoma with penindimoma? I've never heard of that. Like, and the amount of medicines that might treat that are not going to be that much either, right? Those are probably not necessarily going to get solved in the next 20 years. But like general colon cancer that impacts so many people, lung cancers that impact so many people, we know much more. Breast cancer obviously is, in a way, is a success story of where some of the molecular advances and the discoveries of Herceptin and the discoveries that, so Herceptin is made against a specific cancer gene called HER2, HER2. Herceptin's the drug that treats breast cancer patients whose tumor has an alteration in HER2. That was discovered in the late 80s, early 90s. Up until that point, there's no one, before people knew HER2 existed, there was no test for HER2 because they didn't even know it existed. So, and then there was no medicine, even if you had a HER2 change anyways. So what does that do? So those changes of discovering the HER2 gene, developing a test to see if you had it, and then if you had it, developing a medicine specifically for that put those patients into kind of a deep remission that was not possible without that. And if Herceptin as a medicine had never been invented until today, those patients would be some of these patients that did not have a good treatment. Within breast cancer today, there still is a chunk, a big chunk of breast cancer patients that are called triple negative breast cancer patients. It sounds like some, villain name in a novel, triple negative. And it's called triple negative because there are 3 different common things that go wrong in breast cancer that we have medicines for. And unfortunately, this crummy type of breast cancer doesn't have any of them. So the name of the tumor itself tells you basically like, you're out of, it may as well be like, you're out of luck breast cancer, right? I mean, it's just, but that has also made people, because breast cancer is a general common form of cancer, is more on the ropes. Like we've got more patients responding compared to some of the other kinds. That has people really doubling down to go after triple negative breast cancer and find medicines that are going to convert it into something that goes into deeper remission. That's where this all has to go. So like for lung cancer, right, today, we've got effective drugs for, honestly, about 10% of patients, maybe 15%. Like 85% of patients, they're going to get chemo, they're going to get experimental medicines. Immunotherapy is becoming a miracle drug for about 5 to 10% of lung cancer patients. So when it works, it is a miracle drug. But scientists, we still don't even know enough about why it works in the patients it works in. So the best we can do is give it to everybody and hope that one of these people actually responds. So what we've got to do is just get much deeper scientific knowledge about why some of these cool new treatments do work. And then in the ones that don't, also, kind of reverse engineer it. Like, can we convert the patients that don't respond to immunotherapy? Is there a way to basically not just make a new drug to treat them a different way? Can we actually flip it so that now they respond to the drug that we actually have today? Effectively making the tumor into something that will work with the medicine that we have, as opposed to hoping that we'll discover a new medicine that will help them. So I just got a text message from some other faculty who know me, and they can see my calendar, and they saw that I'm giving a talk, and they're like, you're more than 10 minutes late to the meeting.
I'm sorry. Thank you,
Oh, no,
Dr. Shaw.
I knew it. I knew I was doing that. It's okay.
Well,
this was so great.
everybody would be, it's clapping now. You can't hear them, but if
okay.
it was in the faculty club, we would be clapping.
That's
Thank
okay.
you for saving that
Very nice.
5% and
Sure.
keep on going. We loved your presentation.
We will keep chipping away.
Keep
All right.
going.
Really
Thank
nice
you
to meet all
so
of you.
much.
Have a great rest
Bye-bye.
of the day.
You too. Bye-bye.